So let's look at the left versus right hemisphere. While they are very similar in many ways, there are important anatomical differences. And the first is that the right hemisphere is slightly larger and heavier than the left. The left cortex is thicker. The left hemisphere contains relatively more gray matter. And the right hemisphere extends further anteriorly than does the left. So you can see in this picture here, this is looking at the brain from the bottom. So this picture is upside down. So this is the right hemisphere here. And you can see it extends a little bit further anteriorly than the left hemisphere. And the left hemisphere extends further posteriorly than the right. Okay, just some interesting differences. Now the Sylvian fissure, which is this fissure here dividing the temporal lobe from the frontal and parietal lobe, the lateral Sylvian fissure is longer in the left hemisphere than the right and has a gentler slope. Hence, the temporoparietal cortex ventral to that of the fissure is larger on the right. So if you look, here's the right hemisphere. So it goes up more sharply and it's a bit shorter. So this area here, the temporoparietal area, is larger on the right compared to the left. Okay. Now, the left side of the brain has a larger insula. Now, you remember in this picture, if you will see now, if you come for the brain dissection, you'll see you can actually pull apart that part of the brain. And you can take a look in there. You can see here they use fork-like things to just pull it apart. And the left side is a larger insula. The insula is that part inside there. The left side is a wider occipital lobe. The right side is a wider frontal lobe. And the left occipital horn of the lateral ventricle is longer. And I've put this picture in here just to remind you where the ventricles are. Okay. Now, the left, just more differences, anatomical differences between left and right. The left has a larger medial temporal lobe. The left has a larger temporal planum, or planum temporal. And I've put this picture in here just so you can see where the planum is. This is the left side. You can say this is bigger than the right side over there. Okay. There's another picture, the planum temporal. The right side has a double Heschel's gyrus in the primary auditory cortex. Here's a gyrus, the Heschel's gyrus. Okay. So right side is a double Heschel's gyrus. The right side also has a larger medial geniculate nucleus. Now you remember the medial geniculate nucleus is in the thalamus. And I've put this picture in again. You should be familiar with this picture. It's just to remind you where the medial geniculate nucleus is. And the right has a larger area of convexity of the frontal operculum. This is a frontal operculum over there. Okay. You'll see there's the label, operculum frontal. Now I've put these pictures in because if you read it just like that in the books, it can sound a little bit overwhelming. Now, something that is very important for us to know, we all know that left side is verbal and right side is non-verbal and so on, but we must be very careful not to be too absolute about this because laterality is relative. And, for example, the right hemisphere also has some language functions, particularly your prosody, etc. And the cerebral site is at least as important as the cerebral side. So, for example, frontal compared to parietal areas. And laterality is affected by genetic and environmental factors because we know the more, depending on what activity we do and so on, there might be strengthening of certain parts compared to others, and there's a genetic component to it. Now, you'll read quite a bit about handedness. I've put in 3 pictures of 3 famous people who happen to be left-handed, just for interest sake. And you get 2 populations of left-handers. And if your patient is left-handed, it's important to ask them about this. Are they familial left-handers? Do they have people in their family that are left-handed, parents, grandparents, and so on? So is it a genetic thing? Or are they non-familial left-handers? Because if they're left-handed and there isn't a family history of it that can be clearly identified, it is possible that they're left-handed because there was some brain damage causing the functions to move over to the other side. Okay, so you need to always ask about family history of left-handedness if you have a left-handed patient. You will read about the WADA test, and the WADA test is where there's carotid sodium amobarbital injected into the brain, and the one hemisphere is anesthetized. And you'll read about a lot of experiments that were done with a WADA test, because if they can anesthetize the one hemisphere, they can get a sense for what the other hemisphere does or doesn't do. And you'll read quite a bit about this. But just about handedness and speech again, this is very important, because sometimes there's the assumption that if you're, obviously if you're right-handed, then language is on your left, as it should be. But there's often the assumption that if you're left-handed, then your language is on the right-hand side. And that's far oversimplified. So if we look at these statistics, 98% of right-handers show speech disturbance when the left hemisphere is anesthetized, but not the right. So that makes sense, because most of your right-handers are going to have their language in the left hemisphere. 70% of left-handers show speech disturbance when the left hemisphere is anesthetized, but not the right. Okay, so most left-handers also have most of their speech on the left, like as right-handers do. 2% of right-handers have speech functions lateralized to the right hemisphere. So if somebody is right-handed, don't assume that they will automatically have all their language abilities on the left. But there's a very, very small percentage of people who would have it on the right. No right-handers showed bilateral speech organization, and 15% of left-handers showed significant speech disturbance after either sides were anesthetized. Now the visual system, just the basics of the eye. All right. Okay, this is the eyeball. Okay, you know, you've got the pupil, you've got the iris, you've got the lens of the eye. The retina at the back here, this is what is important to us, because the retina forms part of the brain. Okay, and that information goes through to the optic nerve and into the brain. Now, how the eyes work and the visual fields and the swapping and the crossing and so on is very complicated. And while you can try and work it out each time, you know, if you have a question about, for example, let's say somebody has lost vision in the top left visual field, what is wrong with that person's visual tract? While you can try and work it out each time, I found I should just try to learn it off by heart. It was a little bit easier than trying to work it out each time. But so, for example, let's say this is the left-sided right lower quadrant. It's green. Okay, so that would go into that area of the retina, the back of the eye, travel along the optic chiasma, into the optic radiations, and through to the visual cortex. Different to, for example, let's say this area, which is the right eye, left upper quadrant, which gets reflected there, comes along here, comes along there, relays in the right geniculate nucleus, and goes along there and to the visual cortex at the back. All right. Now, this here, the optic chiasma, that is where they cross. Okay. And if you look in the brain, if you actually open up the brain, it's quite easy to see that structure there. Just to look at it from a slightly different perspective, so light falls onto the photoreceptors at the back of the retina, goes into the optic tract, relays in the thalamus, the lateral geniculate nucleus of the thalamus, radiates this way through the optic radiations, and then into the back, into the occipital cortex. Okay. Now, while we're talking about these different systems, I'm also going to be talking about what if there's a disconnection between the left and the right hemispheres. To what extent do the pathways cross, and if they cross, where do they cross? Okay, so that's what I refer to when I talk about disconnection. Now, verbal material is more accurately perceived when presented to the right hemisphere and processed in the left hemisphere. Okay. Visuospatial input is more accurately perceived in the left hemisphere and processed in the right hemisphere. Okay, so I think that makes sense. We know the left side is more verbal, the other side is more nonverbal. So let us look at the auditory system. I'm sure you're all familiar with the basic structure of the ear, so I'm not going to go into that right now. These are the auditory pathways. Information comes in, let's say here through the left ear, to the cochlea, goes to the cochlear nucleus over here. Now watch what happens. Part of the information stays ipsilateral along there. Part of the information swaps over to the other side, to the superior olivary nucleus, goes up to the inferior colliculus. Remember, here, there's the superior colliculus, there's the inferior colliculus, so there's the inferior colliculus. Up to the medial geniculate nucleus. The medial geniculate nucleus is in the thalamus, and then through to the auditory processing cortex, auditory cortex. Okay, so you can see immediately that when it comes to hearing, it is ipsilateral as well as contralateral. So the auditory system has both crossed and uncrossed connections. For example, the left hemisphere receives most of its inputs from the right ear, but also receives input from the left ear. So words played into the left ear can travel directly to the left hemisphere, or can go to the right hemisphere and then to the left hemisphere through the corpus callosum. Okay. The motor system. I know I'm going through these quite quickly, but as I said, we have quite a lot to go through. All right. When we think about the motor system, we've got to think about the basal ganglia. So, and you know that this is the basal ganglia, this picture of the basal ganglia. There's one on the left, on the right. This pinkish part here is the thalamus. Okay. Now, damage to the basal ganglia can lead to problems like Huntington's chorea, Parkinson's disease, and Tourette syndrome, which are all motor control sort of problems. The motor area, this is the primary motor cortex, and the premotor cortex is obviously very important. And disturbance of motor functions would lead to fine movement difficulties, speed and strength difficulties, movement programming problems, problems with voluntary gaze, corollary discharge. That is when, if you're moving, let's say you're driving in a car, let's say you're driving on the back of a bakkie or something, it's bumping up and down, your world is still standing still, even though your whole body is going up and down. Okay, that's because of corollary discharge and speech. Why you would have problems with speech is if you think of where Broca's area is, okay, it's over there. One of the difficulties with Broca's area is that it involves the motor areas, with speech is also a motor difficulty, a motor process. Okay. All right. Now, disconnection of the motor system, which side works with what and where do they cross and so on. The motor system has crossed connections, and they cross in the brainstem. Okay, so if you have, for example, a stroke on the left-hand side over there, it's the right side of your body that will be affected. Now, the somatosensory system is crossed, so sensations of touch in the left hand travel to the right hemisphere and vice versa. Here's an example of somebody who has contralateral neglect. Here. A normal person would draw it like this, but this is actually the model from which the person drew it. So they were asked to draw this, and they drew that. They were asked to draw this, and they drew that. They were asked to draw this, and they drew that. This is a person with contralateral neglect. Okay. People like this also tend to ignore the left side of their bodies. So they'll walk to a door and knock their hand on the side of the door, that kind of thing. Okay. The gustatory system has to do with taste. Now, our children are still being taught at school, well, at least mine were, that the tongue has these different areas, some for sweet, some for sour, some for bitter. It's actually not true. And you'll read this in Colvin-Wishaw, each little taste bud has its own little sensors for sweet, sour, bitter, all the different tastes. So this is what here's the surface of the tongue over there. This is what the little taste bud looks like, or the gustatory taste cell. And the information goes here into the supporting cell, into the sensory nerve fiber and to the brain. So you can see, like this one is for sweet, that one's for bitter, that one's for salty or for sour. Okay, so the idea that we have different areas of our tongues responsible for taste is actually a myth. So we've discovered. Okay. Now, the information coming from those neurons go along the glossopharyngeal nerve as well as the facial nerve. There's synapse in the medulla oblongata of the brainstem, and synapses again in the thalamus, because almost everything relays in the thalamus, and then through to the gustatory cortex, which is in the insula. It's that area there. If you take the brain, you pull it open on the side there, there's that cortex inside there. That is the insular cortex, and it's our primary gustatory area. Okay. The olfactory system. Here's a picture of the nose, the smell system. Here's your nasal passage. Here's your olfactory bulb, that the information from there goes onto the olfactory tract, which goes to the cerebral cortex. Okay? But you have these little fibers growing through here, little neurons growing through here, into the nasal passages, picking up on the chemicals of the foods that you've been eating. Or not only the foods that you've been eating, because you know that there's a big connection, there's a big gap really between the back of our noses and our mouths. Now, when we eat, the particles from the food also goes down nasal passages and gets picked up, and that is part of, so it's not just the smell of the food that we smell, but also our nasal passages have to do with taste as well, and together they form what we call flavor. But of course, also if you smell something directly, then it's these particles, these little neurons that pick up on smell. The skull bone here has these little holes in them called the cribriform plate that the little neurons grow through. Now, here again, here are your little neurons. They go into the olfactory bulb, into the olfactory tract. Have a look here. It goes through to the amygdala, which is why we say smell has a very emotional component to it. When you smell something familiar to you, you can have very strong emotional reactions to it. It might be something that makes you so happy, or makes something that makes you feel frightened or disgusted. And of course, relaying into the entorhinal cortex and then into the hippocampus. So hippocampus we always associate with memory. We tend to have very good memory for smells, and particular smells that have an strong emotional component to them. Now have a look over here as well. There's a synapse there, and then it goes through here through the anterior commissure. Now remember, the corpus callosum isn't the only part of the two hemispheres that are where the two hemispheres are joined. Also at the anterior commissure. So now information from the olfactory tract crosses through the anterior commissure to the other hemisphere. Okay. This is just a picture just to remind you that smells coming into the nose goes to the amygdala, it goes to the limbic area, meaning that it has an emotional component to it. Now, the olfactory system is not crossed. Okay, we know many of our systems are crossed. The olfactory system is not crossed. So input from the left nostril goes straight to the left hemisphere, and vice versa. Input from the right nostril goes straight through to the right hemisphere. But fibers traveling through the anterior commissure join the olfactory region. So eventually that information does get to the other hemisphere, but it's through the anterior commissure. But the system is not crossed.